Hello everyone and welcome to today's live broadcast, The Future of Syphilis Testing, Improve Your Workflow with Novel Technologies. Presented by Dr. Belinda Yen-Lieberman, Dr. Alan Wu, and Dr. Vera Tesic. I'm Christina Jewell of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Bio-Rad Laboratories. Now before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window. Type your question into the box that appears on the screen and we'll answer as many questions as we have time for at the end of the presentation. We will also be conducting a few polls. The polls will pop up in the middle of your slide window. Just check the box that best describes your answer and click Submit. In fact, here is our first poll. What algorithm does your laboratory currently follow or use? Is it traditional algorithm, reverse algorithm, or other? Thanks for your participation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have any trouble seeing or hearing the presentation, just click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Just click on the button at the bottom left corner and follow the process to obtain your credits. I would now like to introduce our presenters. Dr. Belinda Yen-Lieberman is a professor of pathology at the Cleveland Clinic Lerner College of Medicine. She's board certified in clinical virology. Dr. Yen-Lieberman received her Bachelor of Science degree in microbiology from Southern Illinois University and her Master of Science and doctorate degrees in immunology and biochemistry at the University of Arkansas Fayetteville. She completed fellowships in clinical and cellular immunology at Case Western Reserve University and the Cleveland Clinic Foundation. Dr. Yen-Lieberman has authored over 130 articles for numerous medical journals on a wide range of topics involving viruses and infectious disease. Dr. Alan Wu is Chief of Clinical Chemistry and Toxicology at San Francisco General Hospital, Professor of Laboratory Medicine, University of California, San Francisco, and Medical Director for the Pharmacogenomics Laboratory. He received B.S. degrees in chemistry and biology at Purdue University, West Lafayette, Indiana, and a Ph.D. degree in analytical chemistry at the University of Illinois Champaign-Urbana, Illinois. He completed a postdoctoral fellowship in clinical chemistry at Hartford Hospital. Dr. Wu has over 400 publications in peer-reviewed journals. He has also written 5 paperback books consisting of short stories designed to promote the value of the clinical laboratory and pharmacogenomics to the general public. Dr. Vera Tesic works as an assistant professor of pathology and assistant medical director of clinical microbiology and immunology laboratories at the University of Chicago Medicine, Chicago, Illinois. She completed her training in pathology at the University of Illinois, Chicago, followed by a fellowship in clinical microbiology at the University of Utah, ARUP Laboratories. Her areas of interest include application of new technologies into clinical laboratory practice and implementation of cost-effective diagnostic tests for infectious diseases with a particular focus on syphilis testing. To read the full biographies on our speakers, please click on their photos in the top left corner of your screen. I would now like to hand this presentation over to our speakers. Welcome, doctors. You may now begin your presentation.
Thank you. Good morning and good afternoon to everyone. Today I will take the next 15 to 20 minutes to provide you an overview of currently available laboratory diagnostic tests from individuals with syphilis infection. Quickly, the outline will go over the old test versus the new test for syphilis diagnosis, and then follow with the CDC's recommendation for the syphilis diagnostic test. Quickly, we all know the syphilis is a bacteria called Treponema pallidum. So it's a spirochete it's a gram-negative bacteria, and then it looks like a corkscrew-shaped cells. So, like I said, so bacteria is very thin. It's 6 to 15 micrometer, but it's only 0.1 micrometer wide. And then usually there'll be debate about the gram-stain, even though it was decided as gram-negative, but not many people still not quite sure. Normally, you cannot see, visibly with regular microscope, so you have to use what we call dark field microscopy. But in my life in the lab, I have never seen one. So everybody can see this on the Google on my picture. Quickly, I'll just briefly remind everyone about epidemiology. As you can see from the next slide, in the 40s, the prevalence of positive syphilis is really high. So it's 500, close to 600 per 100,000 people. So as time goes, because the invent of penicillin, the treatment, so the prevalence went really down until we get to about late 80s and 90s. Now we have 2017. So the positive rate has significantly increased the last many years due to men have sex with men and many other reasons. So the lab where I work, and we do significant number of the screening tests for adolescents and for people in their 20s to 40s, and be able to capture significant number of people totally asymptomatic but serology positive. And then quickly you show this next map showing the prevalence in 2010. As you can see on the let's see, I want to show people is on this over here, the prevalence is less than 0.2 over 100,000. That is that's 2010. When you get back to the next slide, again you can see the prevalence went to 1.0. We are talking about tenfold increment in the positive people with positive syphilis test result. So then 2016-17, and when you check on the CDC website, it has gone up sixfold increment since the last 2-3 years. So it's a worrisome. It's a test that never going to go down again unless we can making sure everyone gets screened. To detect the test to detect the virus of the bacteria, and normally it's microscopy. As I mentioned earlier, dark field microscopy is something in the past, unless maybe in CDC. And then we actually do not do any of the direct immunofluorescence because we never have any sample that you can capture positive, and no silver stain. So serology tests consists of two kinds. First one is non-Treponema, so it's a rapid plasma reagent. What is so it's VDRL, Venereal Disease Research Lab. Both this non-Treponema tests detect the detection of antibody against cardiolipin, which is a protein produced due to high fever. So they are indirect, but they are very accurate in the case will detect acute infection, right in the middle infection. However, there's a lot of false positive result. It's, for years, that's easy to do, so everyone uses that as a manual test. Then the second one is Treponema test. You detect the presence of antibody, IgG or IgM, toward Treponema pallidum. So again, the direct dark field and test, and unless other people still doing that, I do not know. So let's go discuss a little bit non-Treponema test. As I said, you're really not detecting the bacteria. You're detecting the product due to the bacterial infection. And it's very accurate. For many years, it's very cheap. It looks like pregnancy test type of things really fast. However, they're not specific. So it could be false positive when you have Lyme disease, malaria, TB. I remember toxo, people have pregnancy, IV drug. So as a result, you can see the list of non-specific positive could be, sort of giving you a headache. What should we do? They have a cardiolipin. That's why the old test algorithm is to do the non-specific. When it's equivocal or false positive, then you reflex them to a treponema test. At that time, it was very expensive, and then also, it's all manually. So this test is very good. The RPR and VDR is very good to detect primary and secondary syphilis. So this next slide I want to show you is everyone looks like pregnancy test or monospot. So this test is a particle agglutination, detect the presence of antibody against cardiolipin. So as you can see, this is a quantitative test. This test is very important. You can quantitatively detect the presence of the antibody against cardiolipin. So the higher the titer, the more so-called severity of the cases. So you can see this is very tradition. And the next slide also show you we at the Cleveland Clinic we still have this. We hope we have this by 2018 before we bring in the new test. We are still, I joke with things like shake and bake. You shake this thing, you go read the particle agglutination, but it still works. And however, if you receive 100-some sample, it becomes very, labor intense. So let's talk about treponema test. The last many years, detection for antibody against the bacteria become very popular because it is syphilis. Syphilis caused by Treponema pallidum. We need to detect the antibody caused that. And so there are many tests available, and the fluorescent Treponema pallidum antibody absorption test or Treponema pallidum particle agglutination. And that's the test that a lot of people use for many years. But I'm not quite sure it's FDA cleared, but it's very accurate, so we used to send out to reference lab. The enzyme immunoassay, chemiluminescent immunoassay. The last one is the most important, is multiplex fluorescent immunoassay. Is what we are using. A lot of majority of the lab use that because you actually use recombinant bacterial antigen and the code on the beads. It's a Luminex beads, and then you use an enzyme immunoassay to do the test, and then also particle label, so it's increased the sensitivity and specificity tremendous since we brought that in 4 or 5 years ago. So quickly, next slide, quickly review to you about what's traditional test. This is the I said so it's the old syphilis test algorithm. So when the sample come in, you quickly do a RPR. If the spinal fluid, you do the VDRL. The only difference is the concentration of the antigen. And then when it's negative, and then you can report out, at least when I started, they were still using that. It said no syphilis or recent infection. All of us know when you first get infected, it takes 2 to 4 weeks for individuals to produce antibody. So that window period, that could be negative, but that would be false negative. So in this case, if patient has the right, medical history, they have to be followed up, come back to see the clinician. But you go into the left side of the algorithm, when the RPR or VDR is positive, then you have to confirm with when the RPR is positive, confirm with fluorescent Treponema pallidum test. So you can confirm not only the anti-cardiolipin is positive, patient has fever, inflammation, but this is caused by Treponema pallidum, the bacteria, which you confirm it's a positive syphilis on the left side. And if it's negative, and then, in the lab we're very cautious. We say negative, we'll say probably not syphilis. So the next slide is really standard serology reactivity on the left side is when the time you get infected, and then you follow the time, and from the x-axis you can see at what time the antibody will be produced, and also the what time the antibody against cardiolipin will be available for us to be tested. So there's, again, you see a window period. So there are a lot of times individual could be asymptomatic and then walking around not knowing for a while. If you're young, healthy, and then some of my friends practice in other hospital say sometimes they're walking around, they look perfectly fine, physical does not show. Then when they do the test, they'll find them RPR positive. So in those cases, this is a test that's very crucial. If you don't offer the patient test, and then you could have missed someone who is in the process to convert to positive. So the RPR is important. Confirmatory is as important. So now, just some sort of positive, negative sort of characteristic of this test. The old traditional test is very cheap, low throughput, because you're using your finger making little circles for the agglutination. It's very manual. If every day, like we have sometimes, we have 130, 140, and you can be really labor intense, then it's reliable for detection of active infection. That's really positive. And the RPR interpretation is highly subjective, because I mentioned that it cross-reacts when you have other fever due to herpes, toxoplasma, or even pregnancy. So that could be misleading. And that will, in this case, our clinician here would just ask for come back, retest, and to see if the results still persistent. So reverse, that's the new reverse sequence algorithm. This is why you take the patient sample, you test for the Treponema pallidum antibody test. So it's high throughput, it's automated. So we use BioPlex. There are several other automated EIA platform people are using it. So this test will detect early syphilis and the syphilis being untreated, which is very advantage. And then objective interpretation, because the antibody, if positive, it is against the syphilis bacteria. So the issues of discordant result, I will show you some example. I'm sure from the other lab, from the Dr. Wu or Dr. Tesic's lab, we get things like that, and then you scratch your head and you do some kind of repeat. So the reverse sequence, you do right away. Where I am, we do the syphilis IgG right away. And when it's non-reactive, again, no syphilis or very recent. When you go to on the left side, the algorithm, you see it's reactive. Then you confirm with RPR. Why do we confirm a positive antibody test to syphilis bacteria with RPR? Because RPR test can be done quantitative. So if patient is positive, they've been treated with penicillin, and then they get periodically come in and get a sample and get retested, see if the penicillin is working. That's when you will observe titer slowly going down, so instead going up. So the patient's on the right track, a proper treatment. So it's very important to tell the doctor at what stage your patient is after they've been treated. So if RPR is positive, the syphilis antibody is positive, then the result is positive syphilis. Then, but if the syphilis antibody test is positive, RPR negative, and you have a question mark, it could be someone just have a low immune response, they don't make enough antibody, or whatever reason, or they could be false negative if it's right at the cutoff. So at the Cleveland Clinic, whenever we get positive syphilis test, negative RPR, we go right to a second syphilis, FDA cleared syphilis antibody test, and then just sort of double confirm the second Treponema test with the first name. And they are two different manufacturers, and then they are a little bit different in the sensitivity. The second test is more sensitive, and then the first test is more specific. So here we talk about discordant result. You would think such a simple infection, you get a lot of discordant result. Someone's positive treponema, negative. I just mentioned that to you, and then maybe they're treated, and maybe they have latent syphilis, and then also early infection with syphilis. The antibody is not around yet, and also there could be many cases false positive Treponema test. I'll be quick. We have an example. So we have a 83-year-old woman and patient in the nursing home, and when they test her, she was positive, but she was a little bit above the cutoff. So our ID doctor is really worried, because she's in nursing home, and what would you do when you see this? And you want to retest, so we retest. She was still positive, and so finally the clinician actually, to make things, she's not quite sure that's sort of related with active sexual activity or things like that. So she just went ahead, treated the patient. Then we followed the patient as time went, and actually we saw the RPR number. There was negative, but it was always stayed negative. But the positive, the Treponema test slowly went down after a year or two. So sometimes we in the laboratory can use this will be our tool to repeat the test, redraw, and check other, medical history can help to, sort of resolve the conflict result. So the pros cons, I think this is repeated. We already mentioned, inexpensive, low throughput, manual, and but it's reliable if it's active. And then RPR interpretation is subjective with bearing in mind, so make sure patient doesn't have the other infection. And then reverse, the new test algorithm is high throughput, less manual, actually cheaper in our case, and then can detect early syphilis and the syphilis being untreated. And the objective interpretation, because the test, the antigen were all recombinant, so it's less cross-reactivity with other infections like herpes or toxo. And then there are a lot to do with patient management on this discordant result. I'm sure both Dr. Wu and Dr. Tesic both have the same cases like we have. So now there's got a new comparison. So in United States, a lot of hospital public health still use the old algorithm because it works. Also, the volume is not high, so they will use the RPR. And then, like in our place, we do hundreds, so we use the reverse RPR. But you can see the accuracy. Then there's a third one, the European algorithm. From what I was told, the European algorithm, they use two Treponema EIA tests. They use two, one after another, and to increase the accuracy. So that's also good, maybe more expensive. So quickly, hopefully I'm not taking too long, but quickly. So the CDC issued recommendation in end of 2015. They recommend non-Treponema tests being repeated in 2 to 4 weeks for the patient at risk for primary syphilis. And secondly, the second test should be a Treponema pallidum, the particle agglutination, and that's for those times before we found out a second Treponema syphilis antibody test, we would send it out. And then now we use the second FDA approved. And so this is whenever discordant, you use the second one. So the patient can have 2 syphilis tests and 1 RPR. The non-Treponema test is recommended to monitor patient treatment because the titer of the RPR can change. If someone titers 164 can eventually go really down. That actually indicate to the clinician that patients are making progress and recover. And so the last question, is there a way to have both Treponema and non-Treponema results simultaneously? So for the past few years when I was still full-time, lab director, I keep thinking, they are doing this look like I call it shake and bake. You smear them, look like pregnancy test. You weigh very labor intense. Then you go get a tube. You do the second test. So I think, and then I think the audience will be pleasantly surprised. It is coming, and actually it's FDA cleared. And here's the machine we use. It's random access, and it's easy to operate. It's complete automated. So quickly, the syphilis remains a diagnostic, and diagnosis requires combination laboratory test, and of course, keen clinical, observation. The reverse sequence test, syphilis tests, have key advantage over the conventional one. And in the early phase of implementation, like what we did, we must be prepared to educate and inform a clinician, because we actually gave the talk in both pediatric and adult infectious disease, and we invite them to ask questions. And the laboratory must be open to altering the test algorithm to best serve the clinician. Well, I have a couple of friends working in a smaller place. They actually use the traditional syphilis test because it works better for them, and their volume is very low, which I agree with them 100%. So you have to be very flexible to work with the clinician and your patient population to not to do unnecessary tests. Lastly, the additional study compare the different Treponema assay are barely needed. So this is the end of my talk, and now I'm turning this to Dr. Wu.
Well, thank you, Dr. Yen-Lieberman. So my job is to talk about the BioPlex assay, both the total and RPR, as part of the clinical trial that was submitted to the FDA and received 510K clearance. We were one of the trial sites, and so we have quite a bit of experience with this particular assay. So as Belinda indicated, she uses the BioPlex 2200, which is a automated immunoassay system that uses different colored beads, microbeads that are labeled with fluorescent dye. Each bead color is detected by one color of the laser, and then these are fluorescently labeled so that you can detect the presence of individual analytes. And so this particular instrument allows for simultaneous detection from a single set of beads. So the total syphilis and RPR assay specifications are listed here. It's the 2200 BioPlex analyzer. It uses for the Treponema assay a fusion protein. For the RPR, it's a cardiolipin antigen, this mitochondrial lipid membrane component. There's also an internal standard bead and a serum verification bead. The conjugate is a monoclonal anti-IgM and IgG and IgM antibody, and as a result, you'll get three results. You get the quantitative total anti-treponemal combination IgG and IgM result. You get the RPR, and you get the RPR titer. This is a diagram showing the different beads. The blue beads, as shown here, are the fusion protein for total syphilis, again, both IgG and IgM. The anti-cardiolipin antibody is detected using the RPR, the cardiolipin antigen here. They're all mixed into this cocktail along with the internal standard bead to stabilize or to standardize the detector response, and a serum verification bead just to verify that in fact the sample that you're dealing with is serum. The intended use statement submitted to the FDA is a qualitative test for syphilis diagnosis. It's not intended for screening blood or plasma donors. A second qualitative step, and either the traditional or reverse algorithms for syphilis detection is used, can be done in either way on the BioPlex. And then in order to get a quantitative level, there can be up to 5 dilutions that can determine titers up to 1 to 64. These are the various features. The sample type can be serum, can be plasma, either with EDTA, lithium, or sodium heparin. Very small sample volume of 10 microliters. The results here, qualitative, but you do see an antibody index result for the syphilis test, the reportable range of 0.2 to 8.0 AI, and the RPR also with dilutions up to these 5 different titers, 1 to 4 through 1 to 64. And the RPR signal is ranges of 0.2 to 10 antibody index. The interpretation is given here for the total syphilis. There is a reactive zone of greater than or equal to 1.1. There is the non-reactive zone of less than or equal to 0.8. And there is a equivocal zone. Any values between 0.9 and 1.0 are so listed. And for RPR, anything that is less than a 1.0 is considered non-reactive, and greater than that would be greater than 1.0, greater than or equal to 1.0. So here are some data on the RPR titer interpretations, the various 5 onboard dilutions that are performed, and the final result, as indicated here on the far right, would be the titer that produced the last reactive result. So for example here, if we have a value of 1 to 32, then that would be the last dilution that would be reactive for that particular specimen. The FDA trial consisted of 3 clinical sites, 2 internal sites, and 1 at the manufacturer. Samples collected in 2008, and the predicate devices were the Treponema assay, the TPPA, the manual RPR, and the EIA assays compared to the BioPlex. Approximately 1,900 samples were tested on the BioPlex 2200, Syphilis Total, and RPR assays with results and concordance evaluated. These were broken down into samples that were collected prospectively, that is to say, tests that were ordered on either apparently healthy individuals, pregnant women, HIV patients, and also a set of 540 samples that were retrospectively collected, which included both positive and clinically diagnosed syphilis. The prospective study, the intended use population, were those on whom a syphilis algorithm was ordered, or that they had HIV positivity, or that the woman was pregnant. Dr. Yen-Lieberman showed you data on RPR indicating that many patients who have an HIV infection or who have who are pregnant can produce a false positive result for the RPR, and this was why those particular groups were especially highlighted in this study. So here are the raw data, the prospective samples for those on whom tests were ordered. You can see the BioPlex 2200 here in the top, and the syphilis total, and on the bottom, the RPR. Because the syphilis has an equivocal zone, there are a handful of patients that are in that zone. And overall, against the predicate, the percent positive agreement was 92.5%, and percent negative agreement 97.9% for the total syphilis assay, and 81.5% for the RPR against the predicate, and 96.5% against the predicate for the negative. 81.5% positive agreement, 96.5% negative agreement. Breaking down the groups according to pregnant women, smaller numbers as you can see, but the performance was equal to, and in many cases, superior. And these are the retrospective performances. Again, a smaller subset, those who were clinically positive, many more of them here, and you can see excellent positive agreements, well over 98% in all cases. And finally, the HIV-specific population, again, a population that can produce false positive results on the RPR assay. We see excellent agreement positively at 93% and 94% for negative agreement with the syphilis total assay. So these data confirm the diagnostic utility of the confirmatory tests for both the treponin and non-treponin screening procedures using either the traditional algorithm at 99.8% positive agreement or the reverse algorithm where the BioPlex RPR assay is the first assay done in the reverse algorithm, and that was near 96% positive agreement. As a laboratory, you have really your option of doing whichever procedure that you feel is best useful for your institution. This is the comparison of the two algorithms, the traditional on the top where RPR is done first, then followed up by a predicate test, either the TTPA or the BioPlex syphilis, and the percent agreement here are shown at 99.8% for a positive result and 87.5% for a negative second test. If you do the reverse algorithm, whether you use the TPPA first as your screening test, followed by RPR and confirmed by the BioPlex RPR, or you do the EIA first, or chemiluminescence immunoassay, followed by the RPR. Again, excellent agreements, particularly for the positive TPPA and positive RPR. The precision of the assays on the BioPlex 2200 is excellent, somewhere between 2 to 6% for within and 2 to 10% for between imprecision. This particular assay was tested against a host of different potential interference. This slide here shows the typical interferences that we do in the laboratory: hemoglobin, bilirubin, lipids, proteins, showing really no interferences with these assays. And a more specific interference test against cross-reactivities to other infectious disease serologies, as you see here, CMV, the anticardiolipin antibodies are not directed towards the ones in the RPR, but are directed against those that are produced in autoimmune disease. And again, you can see rubella, very little cross-reactivity to other serologies with the total and RPR BioPlex 2200 assay. So the conclusion for the FDA was that this BioPlex assay shows substantial equivalence to other commercially available predicate assays, and the assay was approved by the FDA and is now available for use. Now, I just have a couple of additional slides of some data that we have generated from this trial. This is a chart of a series of individuals, 9 individuals, on whom there were multiple bleeds. The first line would be the first day bleed, and then the subsequent lines would be bleeds on days 5, 10, 13, 31, etc., after the first bleed. And these would indicate the seroconversion of syphilis over time, and the results of various predicate assays, as well as the BioPlex's ability to detect the first positivity. Now, Dr. Yen-Lieberman indicated that there is this window period here where there can be some assays that are negative, and yet the person is infected and potentially falsely presumed to be not infected. And if you look at the first column here, the BioPlex syphilis, it is consistently either the earliest or equal to, in terms of the earliest, than any of the other treponemal and non-treponemal assays, compared, for example, 14 days earlier then the Abbott Architect, the Trinity Biotech Captiva, which is the IgM assay. The IgG assay, we actually expect it to be later developing because the seroconversion rate is slow. Again, the BioPlex measures both IgM and IgG. The Trinity Assure Trep, and the DSON Liaison. So each of these are either equal to or superior to the other predicate assays. And this other slide, rather complex slide, looks at some of the raw data for 4 different samples. There's the negative control, the positive control, one sample that is weakly positive, and another sample that is strongly positive. Each of these samples were assayed 20 times. There is 100% agreement with the result on all of the samples. And on the second line of each entry is the precision of the antibody index, with the cutoff being 1.0 as indicated at the bottom. And you can see here that when the values are very low, the imprecision is a little bit higher, but still well below 20%, even for the negative control. And then for the positive samples, it's well below 5%. This shows excellent precision that you really cannot get with any of the other manual assays for either treponema or non-treponemal tests. And then this is a dilution guide, so that we are able to onboard do 5 dilutions, and depending on which of the dilution, which of the results of the index is positive, would then equate to a titer that people are used to seeing when it comes to the RPR card. So for example, the third one, if you have a reactive value of 4.0, that would indicate that the titer would be 1 to 8. And then if it's greater than 10, then it's at least 1 to 64. So in summary, the BioPlex 2200 Syphilis Total and RPR assay shows substantial performance equivalence to other commercially tests for treponemal and non-treponemal. I would argue that in some cases it's actually superior in terms of producing an earlier seropositivity. It can be used to test both the RPR and the total assay simultaneously on the same sample, on the same instrument, and allows you to use whichever algorithm that your infectious disease people or your clinical laboratory are comfortable with, whether it is the reverse or the traditional algorithm. I would argue, and I think that what we're going to hear in our next talk from Vera Tesic, that there are advantages of considering the use of the reverse algorithm. So without any further ado, I will turn over the presentation to Dr. Tesic. Vera?
Thank you, Dr. Wu. So, I'm going to talk about our experiences with our evaluation of BioPlex 2200 Syphilis Total and RPR Assay at University of Chicago Medicine. So what kind of prompted us to automate our syphilis testing? There are many benefits of automated platform for syphilis testing, and they are similar to automation of other assays because there is objective and automated screening approach. There is also increased sample throughput, addressed our ergonomic issues, and there is also elimination of observer variability with all manual assays. And with that, we also have reduction of manual labor, and ultimately we have cost decrease. And at the end point, with automation, we also have LIS connectivity, which eliminates clerical error. Besides wanting to automate our syphilis testing, bringing on board BioPlex 2200 allowed us to consolidate testing of other analytes that are available on this platform and replace many assays, either ELISA or immunoassays, IFAs, and agglutination assays that we previously performed on different instruments or manually in our laboratory. The advantages important to us were random access with the additional bonus that control for each analyte is done once a day versus batching the tests and needing to run control each time. This system can automatically process up to 100 samples per hour, and depending on the assays, and first results can be very quickly available, approximately in 40 to 60 minutes. Subsequent patient samples were completely every 30 seconds. So in 2014, we performed traditional testing algorithm. So the traditional algorithm in our laboratory consisted of screening with the RPR, Non-Treponemal Test, that was done as manual agglutination test, and confirmation of reactive results with Treponemal Test in our laboratory was FDA ABS. Response to therapy was followed by RPR titer, usually 6 to 12 months after the treatment or sooner if necessary. And as you can see, in 2014, we performed more than 8,000 manual RPR assays, approximately 700 per month. Our confirmatory FDA ABS was also performed in-house. And we also had 17% false positive RPR tests. In the mid-2015, we switched to the reverse syphilis testing algorithm. Initial screen is for detection of a syphilis IgG antibodies performed on BioPlex 2200 system that simultaneously detects IgG antibodies to multiple Treponema pallidum proteins. Confirmation of syphilis is done with manual RPR assay. In the reverse algorithm, discordant laboratory results, for example, reactive Treponemal test and negative RPR, are resolved with the second Treponemal test, and we switch to Treponema pallidum particle agglutination assay. Again, response to therapy was followed by RPR titer. So as you can see, in 2016, we had more than 10,000 syphilis test requests, and our number of false positive rate dropped from 17% to 10% with the implementation of the reverse algorithm. Increase in number of screening tests reflects this rising tide of new syphilis cases and increased demand of testing. So we can probably expect to see more and more test requests in the future. And with the implementation of the reverse algorithm, we were able to reduce our turnaround time for the first assay that comes out. So with traditional, it took 6.6 hours for the RPR screen to be reported to our patient chart, and now it takes only 2.58 hours for the first assay to be seen in the patient chart. So to summarize, our current syphilis testing algorithm is the reverse algorithm. We screen with Treponemal assays on BioPlex 2200 looking for syphilis IgG and confirm with manual RPR. And we also discordant results resolved with the TPPA. So moving forward, in August this year, FDA cleared the new assay. So the objective of our study was to evaluate this new assay, BioPlex 2200, Syphilis Total and RPR, so which simultaneously detects both treponemal and non-treponemal antibodies and provides RPR titer up to 64. Also detects IgG and IgM antibodies against Treponema pallidum and also non-treponemal reagin antibodies. So basically, laboratories that implement this assay will be able to utilize either traditional or reverse testing algorithm and still have both tests automated with faster turnaround time, increased throughput, and improved workflows. So since we started testing for Syphilis Total and RPR in September 2017, and we tested almost 400 samples. When we compared current syphilis IgG versus syphilis total results, we had 94.1% of agreement for detection of positive results and 99.7% for the negative results. However, when we compared manual RPR CAR test results versus BioPlex RPR, We had 87.1% of agreement with positive results for detection of RPR antibodies and 91.9% for negative results. When we investigated the cause for lower agreement, we found that borderline and weak titers that are 1:1 or 1:2 on manual RPR BD card specimens belong to patients with a remote history of treated syphilis, either or without HIV infection. So detection of positive results in these patients is of little clinical significance. Therefore, we felt confident to remove these specimens with borderline results, borderline titers on manual tests, and our percentage of agreement was higher than up to 94.4 for the acute syphilis that were in either primary or secondary stage. We also calculated turnaround time, and not just for a first syphilis assay, but to complete diagnosis. So current turnaround time is 8 hours, and if Syphilis Total is implemented, it would be 3 hours. This is total of 5 hours reduction that clinicians will get the results sooner. So additional workflow advantages with new assay, when new assay is used, are reduction of manual labor, and we can also dedicate our technology's time to perform other assays. There is better reproducibility and accuracy of results, and what is also important to us, methods are consolidated on one platform, and also there is LIS connectivity for both assays. In conclusion, there is a good agreement with previous assay for detection of patients in primary and secondary laboratory stages of infection. Diagnosis is obtained 3 hours faster, and Syphilis Total and RPR allows both treponemal and non-treponemal assays to be automated, which is a great advantage. Thank you, and we are ready for additional questions.
For your informative presentation. Now, before we start our live Q&A portion of the webinar, we have another poll. Are you considering changing your algorithm in the near future? Just click on yes in less than 1 year, yes in 1 to 2 years, yes in more than 2 years, or no. Thank you again for participating. Okay, now if you have a question you would like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on the screen, and click the send button. We'll answer as many questions as we have time for, but if we are not able to get to your question due to time constraints, your questions will be answered via email following the presentation. Okay, let's get started. Dr. Yen-Lieberman, this first question is for you.
Okay.
Robert would like to know, when you have a TREP positive and RPR negative that is TPPA confirmed, should you follow up with RPR in 2 weeks?
Okay, the question is, if we have the second Treponema test positive, the clinician usually follow up 2 to 4 weeks to retest the patient, making sure there's no false positive. That's mostly routinely for them to follow up. Usually 2 to 4 weeks, that's what I was told.
Thank you. Dr. Wu, this next question is for you. In our laboratory, we tend to have samples that have RPR titers greater than 1 to 64. How would I manage those samples?
Yeah, so right now you'll have to do those dilutions by hand. Bio-Rad BioPlex only allows right now for 5 dilutions and up to 1 to 64. But I do know that they are working on a solution that will allow titers up to 1 to 2,048. So for the moment, we'll have to hold on and do this manually, but that's something that's going to be fixed very, very soon.
Thank you. Dr. Wu, I'd like to ask you this next question. We have an audience member that didn't quite understand the BioPlex RPR dilutions guide slide. You mentioned that at a 4.0 AI, the suggested titer is 1 to 8.
Yes, that is correct. From that table, for the 5 different titers that the BioPlex are able to do online, the maximum titer for a AI of 4 is 1:8.
Thank you, Dr. Wu. Dr. Tesic, this next question is for you. Robert would like to know, is it appropriate to compare an RPR titer versus BioPlex cardiolipid titer particularly?
As you could see with our data, there is a good correlation with BioPlex RPR and manual RPR titers. However, we saw some discrepancy for borderline either weak positive or weak reactive samples, and we could attribute this to difference in method, specifically because there is subjectivity in interpretation of manual agglutination versus something that's done on the machine, and it also has like cutoff that's already, I believe, 0.9. So when you see different methodologies, they will always differ between themselves.
Thank you, Dr. Tesic. Dr. Yen-Lieberman, I'd like to ask you this question. Javier would like to know, is it not necessary to discriminate among IgG and IgM?
We all know when someone is IgM positive detected, which is very rare, that means the infection is recent, and IgG is sometimes for life, forever. So in our experience, we rarely get IgM, and you have to catch the right time. So as a result, the answer is, yeah, there's clinical significance between IgG and IgM. However, in a routine clinical lab, when we do a few thousands a year, we rarely, and we did it for about 3 months, we never have any. So the answer is that, yeah, it's clinical significance, but we rarely have it. Since we are doing Treponema tests at the, as the first, because the reverse algorithm, we are testing anyway, we figure we will not miss anyone who's in the middle of infection one way or the other. Did I get that right?
Thank you, Dr. Yen-Lieberman. Dr. Wu, I would like to ask you this next question. Which method will be preferred 10 years from now, traditional or reverse algorithm?
Wow, that's putting myself on the spot here. I actually think that the reverse algorithm is going to be the way to go, because RPR was really something as a screening test, and it was cheaper and easier. But now that we can do these other tests about the same effort, it seems to me that the reverse algorithm would be best. But I would love to hear that comment from Dr. Yen-Lieberman.
Well, Dr. Wu, you made a good point when you gave your slide showing the Syphilis Total can have both results. So why are we worried? We got both there anyway. So whether you said beautifully, you can, if you like reverse, do it. You don't like reverse, RPR results already there.
Thank you, doctors. Dr. Wu, this one's going to come to you again. Does BioPlex 2200 syphilis work with dried blood spots?
No, not at the moment. I think that's not something that anybody's really interested in. I haven't heard anybody looking to do that, perhaps maybe with samples that are being sent through the mail. But doing these kinds of things is actually pretty difficult. I'm involved with the California Newborn Screening Program, and we use dried blood spots all the time, but not for syphilis.
Thank you. Dr. Wu, I'm going to throw one at you one more time. Is the BioPlex RPR susceptible to prozone effect?
No. That is because the assay is a two-step immunoassay with a wash step in between the two incubations. And so unlike a lot of the other assays, like for specific proteins when we use nephelometry, we don't have a prozone effect here for the 2200.
Thank you, Dr. Wu. Now we have time for one more question, and Dr. Yen-Lieberman, I'm going to give you this one. Herman says, we get a lot of physicians asking the why. as to the discrepant results of the TREP positive, RPR negative, second TREP negative. Is there a standard answer, or just recommend to repeat testing after a period of time? And what is the typical percent of discrepancies?
I don't know what typical percent discrepancy, but all of us offer this test for the past many years get that. So just think about, we'll measure antibody level of the patient toward this organism. It fluctuates. So sometimes, let me give you an example. Someone's taking oral steroids. If they happen to draw blood, they're going to be, the antibody, shortly after they're going to be very low. So it fluctuates. So it's important to tell the patient, since you have a discrepant, to come back, repeat with a new sample. And then in the case, like, are you taking any medication? It's things like that. Also, people post-transplant, they don't have any antibody. They are taking, a drug to build up the immune response. Whenever we get those kind of patients with that background, the clinical picture is very important, and it's always important to reschedule, repeat a new sample.
Thank you, Dr. Yen-Lieberman. And thank you to Dr. Wu and to Dr. Tesic. We have time for just a few final comments. Would anybody like to leave some final comments for our audience? Okay, not necessary. Well, I would like to once again thank Dr. Belinda Yen-Lieberman, Dr. Alan Wu, and Dr. Vera Tesic for their presentations. I would also like to thank LabRoots and Bio-Rad Laboratories for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thanks for joining us, and we hope to see you again soon. Goodbye.
Bye-bye.